Mitosis, sometimes not the most exciting type of cell division to study because it's asexual. It occurs in all cells of the body except for our reproductive organs. In mitosis, the cells are going to replicate, which means to copy their DNA. And they do that because they're going to split apart. So think about this. If they're going to split apart, they need to double everything so that when they split, they are still at full strength. So we call this full strength diploid or 2N, and that means the full set of chromosomes are present. We break this process up into stages. In reality, it's a continuous process, but for learning it, we have the first stage, which is interphase, sometimes called the resting phase, but the cell's not resting at all. It's preparing to divide by duplicating its genetic material. In the next stage, called prophase, we start to see the chromatids. You see, each chromosome is really a pair of DNA strands that we call chromatids. At the same time, there are little centrioles, and their job, you'll find out later, is to pull the chromosomes apart. But at this time, they're just going to separate to opposite poles of the cells. In metaphase, we start getting some pulling because now those centrioles form spindle fibers to each chromosome, and as those fibers shorten, they're going to pull the chromatids to the opposite sides of the cell. Now, instead of just memorizing this, think about this. We've doubled everything, and now we're pulling it apart, and you can just guess that the cell's ready to divide after this metaphase. Anaphase is kind of a continuation. The chromatids are separating, and now we're forming what are called daughter chromosomes because they're at opposite poles of the cell, and they're just copies. So they're kind of the same. Telophase is the last stage, and that's where the daughter chromosomes have arrived, and the cell is going to split in a process called cytokinesis. Cyto, think of cell. And now we have two identical daughter cells. So I sometimes remember this as I polish my apples twice. Interphase, prophase, metaphase, anaphase, telophase. And this is how we grow, right? We all started as a tiny cell. Even Lady Gaga or Michael Jackson or all the famous great people of the world, they all started in a very humble way as a single cell that divided over and over again. Meiosis is perhaps a little more exciting because now we're dealing with sex cells. So we're forming gametes. What a gamete is it's a cell that has half of the set of chromosomes that are normal. And we call this N or haploid. And this makes sense, right? If we're going to make a new individual, then we got to have some genetic recombination or rearrangement of genes so that the offspring are a combination of their parents, but they're not the same as either parent. In a process called crossover, it's a small mistake, really. During meiosis, when the chromosomes are lining up, sometimes a piece of the chromatid may break off along the same place as its neighbor, and those pieces of genetic material exchange when the spindle fiber pulls the chromatids apart. It only occurs in homologous chromosomes, and homologous means they're chromosomes that code for the same thing. And I'll say more about this later. The translocation is a type of crossover, but here we have something more serious because these are not homologous chromosomes. They're not part of the same pair. If you look at the diagram below, in this case, distant pieces of chromosomes are exchanged, and so we have a very severe condition, often serious birth defects, cancer, but not always. It really depends on which chromosome. So think about this. A chromosome pair may have genes that code for the shape of your ear, but a distant neighbor may not have any traits about the ear at all, but they may have traits about the kidney or the stomach. So when you start exchanging genes at that level, then you know you're in for trouble. Spermatogenesis, if you look at that word, Sperm is a male gamete. In Genesis, think of the Bible, the beginnings. So here we have the beginnings of sperm. It all starts in the seminiferous tubules. It's like a long, long spaghetti hose packed inside of each male testes. On the outer part of the wall, if you take a look at the diagram, the outer part of the wall are the young cells. They're called spermatogonium. They are 2N, which means they have a full set of chromosomes, which also means they're not ready to be dispersed from the body. We gotta tear them in half so that they match with the female gamete, and that happens through a process of meiosis, which I've talked about. The spermatogonium becomes a spermatocyte. It's just a little older. Meiosis occurs, and now we have a haploid or N gamete called the spermatid. It starts to develop a tail. Now these cells are small and sleek, and they don't have many of their own energy reserves, so they rely upon these Sertoli cells. Finally, they develop flagella, and they can swim off in search of the female gamete. Oogenesis, beginning of the egg. Here, the primary follicle is going to form. Now, these are very, very tiny, and there's thousands of them in an ovary. They form before birth, before the woman's born. The egg or oocyte is surrounded by a layer of cells called the zona pellucida. It's a little protective layer. It develops into a follicle, which I'll say more about. Estrogen is one of the hormones that's going to trigger the first stage of meiosis, and that drives the primary follicle to become a secondary follicle, which is a little larger, and you can see in this example. As estrogen levels increase or rise, they're going to cause that follicle to swell and mature into what's called a vesicular or a Graafian follicle. Take a look at the diagram. You see there's the secondary follicle, and there's a huge open sac around it. And this sac has a purpose. We have a corpus luteum that's going to form. It's that sac, the Graafian sac, after ovulation. Okay, when the egg is released, the corpus luteum is going to remain behind and release estrogen and progesterone, and it's going to prepare the uterus, the endometrium specifically. It causes it to thicken and prepare for the implantation of that embryo, or the egg at this point, really. Fertilization is going to restore that diploid number that I've been talking about. So we've gone from gametes, or N, to a diploid condition, 2N. See the diagram, you'll see that each chromosome has a pair, and each chromosome is made of 2 strands of chromatid, because each parent is going to contribute half the chromosomes. So we have to have chromosomes in pairs so that we can give up one. I hope that makes sense. Heterozygous, think of different, hetero. That's when the gene pairs are not the same. Homo, think of the same, is when the genes are identical or the same. In dominant genes, those are going to be expressed when our body develops. And we often use capital letters when we're doing genetics problems. But if there are no dominant genes, then the genes that are there, the recessive genes, are expressed. When we use the word phenotype, pheno refers to the appearance or physical expression of a gene, and the genotype just refers to genes themselves. So I'll give you some examples of this. Hey, look at these two brothers. One has a recessive pair for the melanin pigment in the skin, and that really changes things. Pleiotrophy, the E is silent in that word. It's a gene at a single locus, which means location, on the chromosome. So that one location may affect more than one trait. A very common situation in the body. Example is HBS. That gene causes sickle cell anemia or sickle cell disease, same thing. But if you have the HbA, notice the capital A there and the small s above it. This is the normal situation for the production of hemoglobin, so that our red blood cells are plate-shaped and they transport oxygen as they should. A sickle cell it's kind of crescent shaped. It's going to clump and reduce oxygen flow and eventually cause death in places where we don't have good medical treatment. Can two normal parents produce a child with sickle cell disease? I think so. If both parents have the recessive trait for this gene, take a look at the diagram. You see mom and dad each carry the S recessive gene. When we say carry, that's a genetic term. It means that we look normal. Physically, our phenotype is normal, but we carry a recessive gene. And we all have this. We don't all have the S gene, but we all have recessive genes. So the man's phenotype is he's a normal carrier. So on a test, this is just a hint, some test questions will say, what is the phenotype? And if you put AS, no, that's a genotype. Those are the genes. Phenotype is a physical expression. Here's how I work out the problem. The man here I have on top, the arrow indicating the mobility of sperm. If you recall, during meiosis, the chromosomes are going to split. So a gene on one chromosome splits from the gene on the other chromosome. So we have A and S separate. And the female on the far left, it just becomes a matchup. Which egg joins with which sperm? And it's sort of a random process. We don't know for sure, so we work with percentages. Here it is. There's a chance that the gamete big A is going to match up with big A on the upper left, and so forth. And so there is a 25% chance that there's going to be a child with sickle cell disease or sickle cell anemia. Not all recessive traits are bad. Some have, in fact, many have survival value, at least historically. Even sickle cell disease. In places where there is a blood disease called malaria, people with the heterozygous gene pair, AS, or sometimes we call it AA, have immunity to malaria, and they don't suffer from the serious symptoms of sickle cell. So that's the perfect condition. Well, at least hundreds of years ago, that was the perfect condition to have. Take a look at the malaria map and sickle cell. They sort of overlap. So it's not an accident, and it's not just a random thing, this sickle cell disease. And so people with a homozygous recessive sickle cell, they may survive malaria, but they're going to die of sickle cell disease unless they receive extensive medical treatment. But on the other hand, if you have a homozygous normal blood, don't let me lose you here, you will have no natural protection against malaria. And so, okay, you don't die of sickle cell, but you die of malaria if you're in that region. Good example, Peter Muga, who I worked with for 2 years, he was heterozygous. And so he would come to work sometimes, and he'd be achy and complaining, and I'd say, "Hey, Peter, what's up?" And he'd go, "Oh, Nina Sikim Baya, Nina malaria." I go, "Oh my gosh, Peter's got malaria again." And he just felt like he had the flu. Whereas if I got malaria, I would be on my back in a hospital bed with an IV in my arm, fighting for my very life, because I had no protection against malaria. Pedigree, not just for horses and dogs. It's a way we can track genotypes over many generations. There's people out there, that's their full-time job as geneticists, looking at the physical expression of genes. We often use a square to represent a male and a circle for females. And I know earlobe is not very exciting, but we can't look at some things like eye color. My gosh, it's so complicated. Hair color, skin color. But earlobes are pretty simple. One pair of genes. The dominant phenotype is going to be detached. Detached is the dominant. And so the only way to get attached earlobes, that means you don't have a fleshy lobe, is if you have homozygous recessive, 2 little e's. Don't let me lose you. If I'm losing you, then just get out some scratch paper, take some notes, and work through this. Okay, one's not better than the other. if you have detached, it's not better than attached. How will we ever work out this problem? Man with attached earlobes marries a woman with detached. Who cares? Well, think of it as maybe what if this was a deadly disease? Then maybe we do care. So let's figure out how it's done. The way I do is I always look for the homozygous recessive. And if I have those, then it's pretty simple. Believe it or not, it really is. So I'm going to look for attached earlobes. Those are recessive. Ha, I see it, number 3. What is her genotype if both of their daughters have attached earlobes? Aha, so that means the bottom 2 circles, those are daughters. They have homozygous recessive 2 little e's on both of those. How do you get a daughter with 2 recessive little e's? Well, each parent had to give one of them, because remember meiosis, the splitting of a diploid cell into 2 gametes. So half came from mom, half came from dad. So now we know what the 2 parents had in the second generation, and then we can just work backwards. And here it is. I hate to give you the answer, but what you could do is take a look at this and flip back to the previous slide and do it till it makes sense. And pretty soon you'll see it's really fairly simple. Summary here, mitosis, asexual, so we're producing somatic or body cells that are diploid or 2N. Several phases. Interphase is the preparing to divide. Prophase, those chromosomes are going to condense and we see the chromatids. Metaphase, they're going to line up on the equator and the spindle fiber is going to start to tug at those. And then finally anaphase and continuation. Telophase is when they're going to divide. I polish my apples twice. Meiosis. It's similar to mitosis. There's an interphase, prophase, metaphase, anaphase, telophase, but the result here is gametes, because we're not going to double our chromosomes the second time around, and so we get a haploid condition called N. Sometimes there's a crossover where neighboring chromatids break and exchange their material, but a more serious one is the translocation, where non-homologous, that means distant chromosomes exchange, and that is severe. The beginning of sperm, seminiferous tubules, those are like the long hose in testes, where they start out as spermagonia, become spermatocytes, finally spermatids with tails, and during this process they're nourished by the Sertollis. Beginning of the egg, primary follicle develops into a secondary, and this all happens in the ovary. The oocyte surrounded by a zona, and it becomes a follicle. Estrogen is the hormone that moves this process along. When the graphian releases its egg through ovulation, then the corpus luteum is going to pump estrogen and progesterone to prepare the uterus for implantation of the egg. Homozygous, the same. Heterozygous, different. Genes on the chromosome. Dominant, recessive, referring to the expression of the phenotypic expression of the genotype. Pleiotrophy, you have one gene controlling several traits, such as the HBS, and the sickle cell, which has survival, and many recessive have survival value in certain conditions. Pedigree, that is where we track the genes over several generations to determine the genotype from the phenotype we're seeing. And if this doesn't make sense, spend some time with it. It's kind of it's a neat thing, genetics. It's worth learning about.